Welcome back to Click for IB Biology. We're studying genetics and we're looking at monohybrid inheritance. Monohybrid means single gene, and in this particular case we're looking at the situation where there are a single gene, two alleles, and one allele is exhibiting dominance over the other allele. So we're going to look at the patterns of inheritance. There's a blank version of this presentation down in the description. There are 10 questions with answers, and you can use those to consolidate your understanding of the content that we're covering here. So we're going to take an example from the pea, which is shown here. And if you'll notice here that the peas have round, smooth seeds, and these ones here have wrinkled seeds. So this is the characteristic, and our allele key for this is that smooth seed allele, which we're going to call big A, is dominant to the wrinkled seed, which of course we will call lowercase little a. We start with the parental generation, which is designated P, and we write down their phenotype first. So what we're going to do is we're going to cross smooth seeded parent will be crossed with a wrinkle seed parent. Now, of course, these seeds will need to be planted, they will need to grow, they will need to produce flowers before they can actually be crossed in the genetic cross. So that's what we would do. We would take some of these seeds, grow them, some of these seeds, grow them. Once we've got flowers, we would then cross-pollinate, and that cross-pollination is illustrated in this diagram to the left. Now we're going to start with homozygous parents, and so therefore at the gene loci we would have big A, and over here we would have little a and little a. And we try to remember that when we put simple letters like this, that what we're actually implying is a base sequence in DNA which through transcription and translation would result in a protein which is going to cause, by some mechanism, the smoothness of the seed, whereas this one here would produce a protein which by some mechanism, which we don't know, will produce the wrinkling in the seed. So to the genetic cross. So the pollen transfer will go from one plant to the other. Perhaps we should regard this as the male and the female. It's not overly significant. And meiosis, if we start here, meiosis is going to separate the homologous pair, because that's the meiosis 1 with monohybrid is we get the separation, or as they call it, the segregation of alleles of a gene. And so in this case, half of the gametes will be big A, and half of the gametes here will be big A. Okay, so in fact, together, they're always going to be big A. On the other side, we have the separation of the alleles here. So these pollen grains will have big A, pollen grains will have big A. So when the fertilization occurs, this pollen nucleus will fertilize this ovule nucleus, and the new genotype of the embryo would be big A, little a. Now, since half are like that, that would be 50%, or 0.5, 0.5, and so 0.5, 0.5, that's 0.25, the probability of that happening. If this pollen grain fertilizes this ovule, then we would get big A, little a, the heterozygote, and the same is going to be true on the next line. If it were this gamete fertilizing this one, then that would be big A, little a, 0.25 probability, big A, little a, 0.25 probability for both. And therefore the offspring, which are represented in this grid here, all are big A, little a. So all of them, we add the probabilities together, and so they would all be big A, little a. So this box, these boxes here, they represent the probability or the frequency of the gamete's genotype that we would get from the fertilization with these gametes. You perhaps know that this grid is referred after Punnett, Robert Punnett, who was an early geneticist in the 20th century, early 20th century. Now, our offspring, the genotype will be big A, little a, but the phenotype of these offspring is that they would all be smooth seeds. This first generation, the filial generation, is referred to as the F1. So just to summarize the diagram above, P1 times P1 gives us the F1. So the homozygous dominant crossed with the homozygous recessive creates a heterozygous new generation. Now what we're going to do is we're going to carry these heterozygotes forward. So we're going to take two of these seeds, germinate them, and then cross them to do what's called an F1 cross, in which we take smooth seeds and cross them with smooth seeds. So we need the flowers, of course. So the flowers will get the cross-pollination across like this. But we know that the parental genotype in this generation is big A, little a, big A, little a. These are the offspring from the previous cross. If we go through meiosis, we separate the homologous pairs, so the gametes would half be big A and half would be little a. And that's true on the other side as well, of course. Big A, little a. If this pollen grain fertilizes this ovule, then we would end up with a homozygous dominant genotype. If we cross with this one, we would end up with a heterozygote. If this one fertilizes this one, we would end up with another heterozygote. Notice that we always put the dominant allele first. And then this one with this one would give us the homozygous recessive. So let's write the genotype of our offspring underneath. It was big A to 2 big A little a's to 1 little a. Now this is the ratio, the frequency, the probability from a single genetic cross would be in this ratio here. However, if we look at the phenotype of what is now called the second generation F2, these two would look the same, and so we would end up with 3 smooth. We're predicting a ratio of 3 smooth to 1 wrinkled. So in a population of a large number of individuals, the ratio will roughly be 3 to 1, or we could think of this as being a 3 in 4 probability of being smooth and a 1 in 4 probability of being wrinkled. This ratio of 3 to 1 is often referred to, in the F2, is often referred to as the monohybrid ratio. So those are the ratios that you would predict if we were dealing with one gene, 2 alleles, and complete dominance. In the next video, we will look at the inheritance patterns in codominance. There's a chance to subscribe to the channel. That's really appreciated. And here's the playlist with all our genetics videos.